Gene-edited bacon? Scientists have successfully made pigs resistant to a deadly animal virus by editing their genes. Porcine reproductive and respiratory syndrome, or PRRS, causes breathing problems and death in young pigs and stillborns in infected sows. To address this, University of Edinburgh researchers used gene editing to remove a small section of the pig's CD163 receptor, which the PRRS virus attaches to. They then exposed the gene-edited pigs to the virus and found that the animals did not become infected. Scientists say the genetic edit is permanent, meaning the pig's resistance to the disease will be passed down through natural breeding. The Edinburgh team stressed that unlike genetic modification, which is highly controversial, no foreign genes are added in during the editing process. But with no regulations in place, it may take years before the technology can be applied to commercially raised swines. So that GE bacon might have to wait. It's all in the genes. Gene therapy grows new skin for dying boy. A young patient who lost most of his skin to a genetic disease now has a new chance at life, thanks to a breakthrough therapy that gave him replacement skin. The 7-year-old boy was born with junctional epidermal lysis bullosa, a condition in which a gene defect causes the skin to become fragile, often tearing and forming blisters. By June 2015, the boy had gotten 2 bacterial infections that destroyed 2/3 of his skin. He was treated with antibiotics, bandages, and even a skin graft from his father, but nothing worked. Doctors decided to use gene therapy on a patch of non-blistered skin and used a virus to carry a corrected version of the defective gene into the skin cells. The engineered cells were grown into sheets of skin and grafted onto the child's body to replace his missing skin. The grafts took and grew, and the patient was discharged by February 2016. His epidermis is currently stable and doesn't blister or itch.
Designer babies may soon be possible. A team of researchers at Oregon Health and Science University have successfully performed the first known attempt to create genetically modified human embryos. Gene editing using CRISPR technology is capable of finding a target DNA sequence and replacing it with a desired sequence. Once injected, the guide RNA in the CRISPR-Cas9 system seeks out the target DNA before the Cas9 enzyme binds and cuts it. The void in the DNA strand is then filled with the desired sequence. CRISPR technology theoretically makes it possible to modify the genomes of any living thing on Earth. Researchers in China have previously conducted trials using CRISPR technology in cancer patients, but the results revealed a major problem. The technique can cause unwanted mutations in other parts of a DNA sequence. However, the Portland team is confident that it is possible to avoid such errors. New hope for cancer patients. A panel of experts advising the U.S. Food and Drug Administration has unanimously recommended the approval of a new cancer drug from pharmaceutical giant Novartis. The drug would be used to treat people suffering from leukemia. Tisagenlecleucel is a gene therapy drug used to treat a blood cancer known as B-cell acute lymphoblastic leukemia in patients aged 3 to 25. The drug uses chimeric antigen receptor T-cell therapy, which reprograms immune cells to defeat cancer. The treatment first extracts a patient's own T-cells, then genetically modifies them, using tisagenlecleucel to grow new receptors. These receptors target CD19, a protein expressed by malignant B-cells. Copies of the new cells are grown in the lab by the millions and injected back into the patient, where they then attack the cancer cells. In one test, the treatment led to complete remission in 83% of 52 patients. But it has potentially deadly short-term side effects, such as high fevers, low blood pressure, and lung problems. It's unclear what the long-term side effects are, if any. Therefore, patients who receive treatment are recommended to be monitored for 15 years. The FDA will likely decide whether or not to approve the Novartis drug in the next few months. If they do, it will be the first gene therapy drug of its kind to hit the U.S. market.
Genes can help find time of death. Post-death gene activity may one day be a new area for forensic specialists to explore. A study from Spain has found that gene activity in dead bodies can point to the time of death. It found post-death activity in the brain and spleen to show little change. However, They also discovered post-mortem fluctuations across 600 muscle genes. The research was done via an algorithm that looked at samples from 399 people. The program attempted to tell the times of death for 129. In blood samples, it found that DNA production, immune response, and metabolism declined 6 hours after death. Most gene activity was found to occur between 7 and 14 hours after loss of life. If developed further, their approach could one day be used as a criminal investigation tool.
Identical twins unidentical after one spends a year in space. This is Mark and Scott Kelly, both NASA astronauts and identical twins. Well, at least they used to be. Following his year in space, scientists compared the DNA and RNA of Scott Kelly with his identical twin brother Mark via whole genome sequencing. They found that 7% of Kelly's DNA had changed into so-called space genes. As well as that, his immune system, bone formation, and eyesight all went under changes during his year in space. Telomeres, caps on chromosomes, typically shorten as a person ages. Kelly's lengthened but returned to normal 2 days after he returned to Earth in 2016. So, given a chance, would you spend any time in space? How to edit genetics without changing DNA. Scientific brilliance coming out of the MIT Harvard Broad Institute may one day better the lives of millions. DNA directs protein on how to build cells via RNA. A new study, published in the journal Science, details how DNA instructions can be chemically altered via edits to information in the RNA. The DNA remains unchanged, while proteins build cells based on the edited instructions. The method is not ready for human trials, but could one day help those suffering from degenerative diseases.